Okay, so hello everyone. Today is the final session of this course, Environmental Chemistry and Microbiology. Today I'll discuss all the topics, all the 12 topics that we have discussed previously in brief, and like I've prepared certain questions related to every week's topic. So I'll discuss the question, and along with the questions, we'll keep on discussing and revising the topics from each week. I'll tell you what are the important things to remember for your exams mainly. And if you have any doubt in any of the questions discussed today or any of the questions that we have discussed previously, or if you have any doubt in any of the topics that are there in this course, please let me know since this is the final session. So if you have any kind of doubt or problem, uh, this is the time you can get it resolved. Okay, before starting, I just want to mention, I hope you have seen that the answers have been changed for the questions that you told me for the assignment questions from different weeks. So,
Yes, ma'am. Only week 5.
week
Hello.
one, week 5, question 17, it was correct answer only. Week 1, week 6, week 8, and week 10.
Okay, ma'am.
Okay, week 5, can you tell me what was the question?
Yes, ma'am. All okay.
Uh,
Yes, ma'am.
yeah, could
Uh,
you please tell me the question once?
yes, ma'am. Atmospheric nitrogen fixation is similar to
Huh.
Haber's process for ammonia synthesis, electric arc method for nitric acid preparation, cyanamide process for ammonia synthesis, none the above.
Hmm.
আমরা A করেছিলাম ম্যাম we
Huh.
have done A, sorry.
This would be option B, na, nitric acid synthesis. Like so, the difference, like the main point that you have to focus in the question is atmospheric nitrogen fixation. Okay, the nitrogen fixation that we have studied in the previous session, that was done by the bacteria, soil bacteria. If you remember, we have talked about rhizobium that was uh, living in the nodules of the roots of leguminous plants, and they were fixing the atmospheric nitrogen into ammonia. So that is similar to Haber's process of ammonia synthesis. But the atmospheric nitrogen fixation, if you remember, I have shown you a diagram where lightning was causing the atmospheric nitrogen to convert into nitrate. Okay, so that is what is analogous to the electric, whatever the option was, nitrate synthesis process, basically chemical
Method. Okay,
process.
ma'am.
Okay, so that question was answered correctly. Rest, I have gone through all the questions and I told them to change the answers. So I think they have updated. I checked on the portal itself. They
Yes,
have
ma'am. They
changed
have
the
updated.
answers. So I hope that is.
Ma'am, in week 11, I have to doubt.
Yeah, tell me.
Yes, ma'am. Uh, First question is question number 8.
Hmm.
Alveolar sacs are colonized by dash and alveolar ducts are colonized by dash.
Hmm.
The options were Coaxiella, Coaxiella,
Hmm.
uh, Haemophilus influenzae,
Hmm.
Streptococcus pyogenes, Staphylococcus aureus, Mycobacterium tuberculosis.
Okay.
So, ma'am, which option was correct?
Uh, just a second. I did see. I think last time also you asked this question, right? Just a second. Let me open the question. This is week 11's assignment, right?
Yes, ma'am.
I'm opening my portal. Just give me a minute. All of you have joined right now. We are right now discussing the the doubts that you had in the previous session. There was a list of questions that were shared with me. So some of the answers were given incorrectly uh, in your answer key. So those questions have been updated now. So I hope all of you have seen it. Now we are discussing whatever doubts you have. So all of you who have joined late, if you have any doubt in any of the questions that are there in your assignments or any of the questions that I have discussed with you, please let me know. After that, I'll start the session. I have prepared again some more questions related to all the topics, and then we'll discuss those questions and we'll discuss the topics along with the questions. Okay, why is phosphite rarely found in? Okay, my portal is not getting logged in. Okay, I'll check this. Why is phosphite rarely found in the pleasant environment? What is the answer given to this question? Like, is the answer given wrong? Question number 8 and question number 18. Okay, just give me 1 minute. I'll just switch off my video. It is taking my bandwidth. Okay, the alveolar sacs, I think the option A should be the correct answer. Coxiella should be colonized in the alveolar sacs, and alveolar ducts are colonized by chlamydia. Is this the answer given? Hello?
Hello, ma'am.
Yeah.
Ma'am, in question number 8, they are
Yeah.
showing option B.
Option
And
B,
in
chlamydia.
question number,
Okay.
yes, ma'am.
And question number
And
18?
in question number 18, they are showing option C.
Question number 18, they are showing option
Option
C,
C, ma'am.
lack of enzymes. Okay, that is also wrong. Okay, I'll let them know. In question number 18, it should be rapid chemical oxidation in air. Okay. Present environment. It should not be lack of enzymes. I'll let them know. They'll update these answers as well. Okay. So this will be
Okay,
option
ma'am.
B. Question number 18 will be option B, and question number 8 is it should be option A. Okay.
Okay, ma'am. Okay, ma'am. Thank
Okay.
you, ma'am.
In week 12, answers. Acha, it's not released yet, right?
Yes, ma'am.
Okay. Okay. We'll see. I'll start today's session. So we'll discuss the topics and questions, and let me know if you have any other doubt. Okay.
Sure, ma'am.
Yeah.
Ma'am, other than these 2 weeks, the rest of the week's answers has been corrected.
Yeah. That is what I told. I've mailed them, so they have updated the answers. Only one question number. I think week 5, question number 17. You have mentioned, one of you have mentioned, so that question was correct. So that is what I was explaining. So question number 17, it was about atmospheric nitrogen fixation. It is similar to which of the process? So it is similar to the nitrate synthesis process. So that one was the correct answer only. Rest of the questions, wherever the answers were given wrong, I have asked them to change it, so they have updated it. For these two questions also, I'll mail them, and I'll check the week 12 assignment also, if they're like, once the answer key is released, if there are any error, I'll let them know about those 2 weeks questions also. They'll update it. Okay, let's start with today's session. Question number 1. A 0.1 molar solution of weak monoprotic organic acid is 1.3% ionized at 25 degrees Celsius. Value of Ka is approximately. So all of you do the calculation and let me know the answer. You have 4 options. Option A, 2.8 into 10 raised power of minus 3. Option B, 1.7 into 10 raised power of minus 5. Option C, 5.1 into 10 raised power of minus 5. And option D, 4.2 into 10 raised power of minus 6. Okay. We have already solved such questions in first week session. So try to first and second week session we have solved such questions. So try to solve it. Today is the revision session, so we'll discuss questions from all the topics. Okay. Quickly use your calculator and give me the answer. Okay, I have 2 answers in the chat box. Option B. What about others? Anyone else is able to solve this? None of you are able to solve it, only 2 people are solving it. We have already discussed such questions, right? Your exams are approaching, you should know how to solve this question by now. Okay, I'll give you some hint and then do the rest of the calculation. So we have a weak monoprotic acid, right? So that means, let's say any arbitrary acid, Ha. It will dissociate into H plus and A minus ions. Okay, it's a monoprotic acid, so only one proton is there, so it will dissociate into one proton and one anion. Then it is saying it is our starting concentration is 0.1 molar. So when we are taking this monoprotic acid, the concentration was 0.1 molar and the initial concentration of these two will be 0 because right now the dissociation hasn't started. Once the equilibrium is achieved, so some of the Ha will be dissociated into H plus and A minus ions. So part of this 0.1 molar will get converted into these two ions. And what is the rate by which this is getting ionized? 1.3%. So 1.3% of this original will get dissociated into these two ions. So let's say 1.3% is X. Okay, so that means from the original 0.1 molar, 0.1 X amount will get dissociated into H plus and A minus ion, and the concentrations for H plus and A minus ion will be 0.1 X. Okay, both of them will be 0.1 X. Now how do we define Ka? Dissociation constant. Dissociation constant is defined as the concentrations of the products divided by the concentration of the reactants. So we have 2 products, so it will be in multiplication and then the reactants will be in the denominator. So now we have 0.1 X into 0.1 X divided by 0.1 1 minus x. So 1 of this will get cancelled out and we'll have 0.1 x square by 1 minus x. And you can actually neglect the 1 minus x part because x is 1.3%. 1.3% means 1.3 divided by 100, that is 0.013, and it is very less than 1. So 1 minus 0.013 will be approximately equal to 1 only. So you can approximately write this as 0.1 x square. Now you know the value of x, then solve it. This is the solution. So 4 people have answered option B, 1.7 into 10 raised power of minus 5. That is the correct answer. That is what we are getting. Rest of you who haven't solved this, do you understand the solution now? This is your last chance to ask any questions. If you have any doubt or if you still don't understand how to solve this question, I can explain again. If you want a simpler explanation, let me know. But you have to tell me now if you have any doubt. Is it clear to everyone? Okay. So at the beginning, we had 0.1 molar of the weak monoprotic acid, and that is dissociating by the rate of 1.3%. So 1.3% of the initial will be lost from the reactant and it will be added in the product. And then you have to calculate the Ka value. Ka is the dissociation constant. Dissociation constant for acids are written as Ka. For bases, we write Kb. Okay. Any doubt? Any questions? Okay, moving on to the next question. pH of the buffer solution containing 0.1 molar solution each of NH3 and NH4Cl is approximately. So you have to tell the pH of a buffer solution whose concentrations of ammonia and ammonium chloride is given, and pKb is given for ammonium hydroxide. Tell me how to solve it and tell me what would be the correct answer. Write the correct answers in the chat box. Quickly do the calculation and write the correct answers in the chat box. All of these things have already been taught in the past 12 sessions, so today you all should be able to answer. Okay, I have one answer in the chat box. 2. What about others? All of you write the answers and then I'll discuss. So our concentrations for ammonia and ammonium chloride both are given as 0.1 molar. Okay, I have 4 answers. Anyone else wants to answer? What formula are we going to use? First formula that we are going to use is POH is equal to pKb plus log salt concentration upon base concentration. Okay. So pKb value is given. Salt concentration and base concentration, both of them are equal, so it will be 1 and log of 1 is 0. So once you have POH, the second formula that we are going to use is POH is equal to 14, pH is equal to 14 minus POH. These are the two formulas that we have to use, and you can easily find the solution. What about the other people? I have 4 people answering in the chat box. The others, I hope you are understanding. I have now told you how to calculate it. Can you do the calculation now? Let's see the solution. The solution is 9.26. That is the correct answer. So all of you have answered in the chat box are right. Option B, 9.26. Is this clear? Any doubt, any question regarding this? So similar to this, if if it was a question about an acidic buffer, same formula can be used in terms of pH and pKa. So there are two forms of Henderson-Hasselbalch equation. Either we write POH is equal to pKb plus log salt upon base concentration, or we write pH is equal to pKa plus log salt upon acid concentration. Okay, so these two formulas are there. So depending on the type of dissociation that is happening, if it is a basic buffer, you will be given the pKb value or Kb value, and you have to calculate the POH first. If it's acidic buffer, then pKa or Ka value will be given. If Ka value is given, you can easily calculate the pKa value. So Ka will be equal to minus log, pKa will be equal to minus log of Ka, or pKb is equal to minus log of Kb. pH is equal to minus log of H plus. POH is equal to minus log of OH minus. Why am I telling you all this? Because these are the basic formulas. If you remember this, you can solve any question in this topic. So you have to just remember the Henderson-Hasselbalch equation and basic information about what Ka, Kb means, what is pK, pKb, pH, POH, what is the relationship between pH and POH. If you know these things, you can solve any question. Okay. Moving on to the third question. Decomposition of hydroxylamine, that is NH2OH, in the presence of oxygen follows the rate expression. Rate is equal to K observed NH2OH concentration multiplied by O2 concentration. So this is the rate equation given for the decomposition of hydroxylamine in presence of oxygen. Now the value of K observed is given as 0.237 into 10 raised power of minus 4 liter per mole per second at 0 degree Celsius, and rises to 2.64 into 10 raised power of minus 4 liter per mole per second at 25 degrees Celsius. So the rate constant is different at two different temperatures. We have to calculate the value of EA. You have to calculate the value of EA, that is the activation energy. Okay, so you know rate is dependent on temperature. Rate of any equation, rate of any chemical reaction, it is dependent on temperature. So at different temperature, you will have a different rate and hence different rate constant. So the rate constant at 0 degree Celsius is given, rate constant at 25 degree Celsius is given, and you have to calculate the EA. All of you who are able to solve the questions, please write the answers in the chat box. The equation that you have to use in this question is the Arrhenius equation. We have discussed all these equations and all these questions, these type of questions in the previous session, so you should be able to solve this. All of you quickly solve it. I have 3 answers in the chat box. What about others? Anyone else is able to solve this? Only 3 people have solved it. If you are having any difficulty in solving these questions, please let me know. I can help you out. Okay, let's see the solution. 3 of you are saying option A, 65.2. That is the correct answer. So this is how you have to solve it. So this is the Arrhenius equation given here. K is equal to A, that is the constant, Arrhenius constant, and then e raised to the power of minus Ea by RT. So E is the exponential value. So the value of E is equal to 2.71, around this, and it is raised to the power of Ea. What you have to solve, Ea is the activation energy, R is the gas constant. So the value of gas constant in SI unit is 8.314 in joules per Kelvin per mole. And if any other like mostly the questions will be either in joules or it will be in calories, so you should know the value of gas constant in both the units. And T is the temperature. Here temperature will always be taken in form of Kelvin. So now we'll have two Arrhenius equation, right? We have two values of K given. Here is the one value and here is the second value. So we'll have two Arrhenius equation. If you divide both of the equation, you will get K1 by K2 is equal to e raised to the power of minus Ea by R T1. T1 is the first temperature where the rate constant is K1, and T2 is the second temperature. So plus Ea by R T2. Okay. So if you take natural log both sides, then you will get the equation as this: log K1 upon K2 is equal to minus Ea by R, 1 by T1 minus 1 by T2. So you replace the value. So the value of T1 and T2 will be taken in Kelvin. So we had in the question 0 degree Celsius and 25 degree Celsius. So 0 degree Celsius will be 273 Kelvin and 25 degree Celsius will be 273 plus 25, that is 298 Kelvin. Okay. So this is the values that we have taken, and then you can solve for Ea. Anybody has any doubt in this question? Any doubt, any questions? If you are having difficulty solving this question, solving these type of questions, You can ask me that also. I can help you out maybe. If you tell me what your doubt is, then only I can help you out. Is it clear to everyone? Okay. So once you solve this, you will get the answer in joules. So 65,000 something something you will get, and then just divide it by 1,000 to get the answer in kilojoule, because the options here given in kilojoule. So just divide the whatever answer you are getting, just divide it by 1,000 to get the answer in kilojoules per mole. Okay. So all of you have answered. Option A is the correct answer. All of you who haven't answered, I hope it is clear to you. Next question is, the unit of rate constant for a zero order reaction can be expressed as. What is the rate? What is the unit of the rate constant for a zero order reaction? How do we find out the unit of rate constant of any reaction? Tell me. What would be the answer? Okay, I have 5 answers in the chat box. All of them are option C. Anyone else wants to write the answer? Option C, liter inverse mole per second. That is the correct answer. So how do we solve these questions? So if any reaction is there, A goes to B, then how do we write a rate for this reaction? Either it will be decrease of uh, the concentration of A per unit time, or it will be formation of B. So increase in the concentration of B per unit time. So this is how we express rate, and this will be equal to K, that is the rate constant, multiplied by the concentration of the reactant raised to the power of N. N is the order of the reaction. Okay. So N is the order of the reaction. Now, whatever is the order of the reaction, depending on the unit of K will change, because the unit of A is fixed, unit of time is fixed. So whatever is the change will be in the unit of K. Okay. So the order of reaction here is 0. So that means this will be minus dA by dt is equal to K, A raised to the power of 0, and anything raised to the power of 0 is 1. So the unit of K will be equal to the unit of A divided by the unit of T. So the unit of A is moles per liter. The concentration is always written in moles per liter. And the unit of time is, the SI unit of time is second. Okay, so since it is in denominator, if we take it in numerator, it will be also per second. So moles, liter inverse, second inverse. So this is the correct answer. If instead of zero order reaction, if I have asked you first order reaction, then what would be the answer? Tell me what would be the unit of rate constant for a first order reaction. Now that you know how to find the unit of a rate constant, what would be the unit of the rate constant for a first order reaction? Anybody? Now you know how to solve the unit of a rate constant, right?
Second inverse.
Sorry?
Ma'am, second inverse.
Yes, second inverse. So time, the SI unit of time is second, so second inverse. Okay. So
Yes, ma'am.
it will be a raised to the power of 1. Instead of 0, it would be 1. So we'll have, so it will be dA by dt is equal to K a raised to the power of 1, since the order of the reaction is 1. Then what would be the unit of dA by dt? It will be moles per liter per second, and here we don't know the unit of K. A unit of A would be moles per liter. Okay, so this will get cancelled out and your unit of K will be second inverse. Okay, similarly if it is 2, then here we will have mole per liter whole square. So your unit of K for a, so this is for a first order reaction. For a second order reaction, the unit of K will become mole inverse liter in mole inverse liter and then second inverse. Okay, so this will go on that side of the equation and then you will have this. Okay, so and for any order reaction, like if I say fifth order reaction, you can find the unit of rate constant for that as well. Okay, I hope that's clear to everybody.
Yes, ma'am.
Okay. So this is just the same thing that I've explained. Next question is, the solubility product of AgCl is 1.1 into 10 raised power of minus 10 at 25 degrees Celsius. Its solubility is. So we have discussed this question also previously. So tell me what would be the answer. The solubility product for AgCl is 1.1 into 10 raised power of minus 10 at 25 degrees Celsius. What is its solubility? Molecular weight is given for AgCl 143.5. So the answer you have to give is in grams per liter. So calculate carefully and then tell me the answer. You have 4 options given. Quickly do the calculation. How do we calculate it? I have 2 answers in the chat box. What about others? Anyone else is able to solve this? So I'll give you some hint how to solve it. So AgCl will, when it is solubilized in the ionic form, it will be dissociated into Ag plus and Cl minus. Okay, so AgCl will be dissociated into Ag plus and Cl minus. The solubility of AgCl, let's say, is S. Then the solubility of Ag plus and Cl minus will also be S, and the solubility product Ksp will be equal to the concentration of Ag plus multiplied by the concentration of Cl minus, raised to the power of 1 because the moles are 1. The stoichiometric coefficient here is 1. So this will be equal to S square. So now you have the value of S here. So you can find the solubility product. This solubility product that you will find, it will be in what unit? It will be in molar unit. Okay. So from molar, you have to solve for grams per liter. 3 people have answered option C. Now I have told you how to calculate. So tell me if anyone else is able to solve it. Now you know what calculation has to be done. Do the calculation and let me know the answer. Even if you think that already the answers have been written and what's the point of writing it, write it for my sake so that I will get to know if you are understanding or not. If you are not understanding something, please ask. I'm requesting you again and again, please ask. Anyone else is able to solve the question? So you have the value of S. So Ksp will be, so you have the value of Ksp, sorry. So you have to find for S. So Ksp is 1.1 into 10 raised power of minus 10. So S will be under root of 1.1 into 10 raised power of minus 10. Once you solve that, just multiply it with the molecular weight, that is 143.5, to get the answer in grams per liter. So your final answer would be option C, 1.5 into 10 raised power of minus 3. So this is how you have to solve. So under root of 1.1 into 10 raised power of minus 10 would be 1.05 into 10 raised power of minus 5. If you multiply it by 143.5, you will get 1.5 into 10 raised power of minus 3. So 1 molar is equal to, so 1 molar means this is 1 mole per liter. Okay, and mole is basically, and molecular weight is basically grams per mole. Molecular weight is equal to grams per mole. So if you multiply this and this, you will get grams per liter, right? Molar into molecular weight will give you grams per liter. Mole and mole will get cancelled out. This is what you have to solve. Okay, this is what you got in molar multiplied with the molarity multiplied with the molecular weight, and you will get the answer in grams per liter. Is this clear to everyone? Please let me know if you have any doubt. So option C is the correct answer. I hope that's clear to everyone. Next question is, THMs are produced by the reaction of. THMs are produced by the reaction of. Option A, Fe2 plus with chlorine. Option B, organic matter with chlorine. Option C, organic matter with ozone. And option D, phenol with chlorine dioxide. What are THMs? THMs are trihalomethanes. Trihalomethanes. Okay. They are produced by the reaction of. Quickly write the answer in the chat box now. You don't have to calculate anything. Just write the answers in the chat box. Trihalomethanes are produced by the reaction of. I have only 2 answers in the chat box. This is a theoretical question. You don't have to calculate anything. At least answer these. Anyone else wants to answer? Okay. 4 of you are saying option B, organic matter with chlorine. That is the correct answer. So I told you the name itself says trihalomethane. THM is trihalomethane. So halo, here halo is the halogen. Okay, halogen. And what are halogens? So all the chlorine, bromine, iodine, fluorine, all of these are called halogens. This group is called halogen group. And so organic matters, when they react with chlorine, they will release trihalomethane. Okay, so methane is basically CH4, right? Methane is CH4. So if you replace the 3 of these hydrogen with any halogen molecule, you will get trihalomethane. So if 3 of these hydrogen are replaced by chlorine, you will get chloroform, that is CHCl3. So one carbon will have one electron will be shared by hydrogen, and 3 of them will be shared with chlorine. Okay, so this is chloroform. Similarly, bromo dichloromethane, that means CHBrCl2. So one hydrogen is replaced by bromine and 2 of them are replaced by chlorine. Or dibromochloromethane. So that means 2 of them are replaced by bromine, 1 of them is replaced by chlorine. So this will be CHBr2Cl, or bromoform, that is CHBr3. So all these products, they are formed by the reaction between the chlorine or bromine and the organic matter during disinfection process. Okay, so these are the products of disinfection, byproducts of disinfection process. Is this clear?
Yes, ma'am. Ma'am, could you please show us the previous slide?
Yeah, this one.
Yes, ma'am. Thank you, ma'am.
Okay. Okay, then moving on to the next question. An acidic buffer solution can be prepared by mixing the solutions of. Acidic buffer solution can be prepared by mixing the solutions of option A, ammonium acetate and acetic acid. Option B, ammonium chloride and ammonium hydroxide. Option C, sulfuric acid and sodium hydroxide. Option D, sodium chloride and sodium hydroxide. How can we prepare an acidic buffer? Quickly write your answers in the chat box. Ammonium acetate and acetic acid, ammonium chloride and ammonium hydroxide, sulfuric acid and sodium hydroxide, sodium chloride and sodium hydroxide. What would be the correct answer? All of you write your answers in the chat box. I have 4 answers in the chat box already. Anyone else? Anyone else wants to answer? What is a buffer solution? How do we prepare any buffer? Forget about acid. Forget about acidic or basic. How do we prepare any buffer solution? What are the 2 components of a buffer solution? Can anyone tell me?
Ma'am, weak acid and its salt with a strong base.
Yes. So weak acid and its conjugate salt that would be prepared by a strong base, or vice versa, weak base and its conjugate salt prepared by a strong acid. Okay, so you need a weak acid and a conjugate base, or a weak base and a conjugate acid to make a buffer. Okay, why do we need a weak acid or weak base? Because they have a slow dissociation in the solution, so they will slowly dissociate into its constituent ion. If you take a strong acid, like here sodium hydroxide is a strong base, or sulfuric acid is a strong acid, if you take a strong acid or a strong base, as soon as you put it in a solution, it will dissociate completely into its constituent ions, that is sulfuric. For sulfuric acid you will have H2 H plus ions and sulfate ions, SO4 2 minus ions. Okay, for sodium hydroxide you will have sodium ions and hydroxide ions. Okay, so that's why you need to make a buffer with weak acids or weak bases. Now let's see the options here. So all of you who have answered, you are saying option A, that is the correct answer. So if you see here, ammonium acetate and acetic acid. So acetic acid is a weak acid, and ammonium acetate, so it is prepared by the by this weak acid and a strong base. So what is the base here? Base is NH4OH, ammonium hydroxide, and acetic acid will give us ammonium acetate. Okay, so ammonium acetate and acetic acid will give you an acidic buffer solution. So if you have this combination, the top combination, you will get a acidic buffer, and if you have this combination, you will get a basic buffer. Okay, this is clear. The second option was ammonium chloride and ammonium hydroxide. Now ammonium hydroxide, it's a base and it's a slightly strong base only, and ammonium chloride is the conjugate salt. If you try to make a buffer out of this, you will get a basic buffer and not an acidic buffer. With sulfuric acid and sodium hydroxide, what will happen? This is an acid base reaction. You will get salt. Sodium chloride will form. Sodium sulfate will form. Okay, and then sodium chloride and sodium hydroxide, we can't make a buffer out of it. Why? Because it's a strong acid. Sodium hydroxide is a strong acid, and sodium chloride is also the salt of a strong acid and strong base. So sodium chloride is made by sodium hydroxide and hydrochloric acid. So sodium chloride is a result of a strong acid and a strong base reaction. So you need a salt that is made from a weak acid and a strong base, or a weak base and a strong acid. Okay, and you need the same acid or base that is used for making that salt. Okay, is this clear to everyone? So option A is the correct answer. Any doubt in this or any questions? Let's move on to the next question. If an atom's atomic number is given by Z, its atomic mass by A, and its neutron number is N, which of the following is correct? Options are: option A, N is equal to A plus Z, option B, N is equal to Z minus A, option C, N is equal to A minus Z, and option D, none of the above is correct. So what would be the correct answer? You have an atom's atomic number given by Z, atomic mass given by A, and neutron given by N. So which relation is correct? All of you write your answers in the chat box. I have 4 answers. Anyone else wants to answer? Okay. I have 5 answers now. All of them are option C, N is equal to A minus Z. So how do we write any atoms atomic number and mass number? So if we have an atom M, so we'll have the atomic number and the mass number written. So atomic number is the number of protons and the mass number is the number of protons plus number of neutrons. Okay. So number of neutron plus number of proton will be A and number of proton will be equal to Z. So P is equal to Z. So we can also write this as N plus Z is equal to A or N is equal to A minus Z. Okay. Is this clear? So neutrons plus protons is equal to the mass number. Number of protons is equal to the atomic number. So option C is the correct answer. Is this clear to everyone? If you have any question, please let me know, okay? Moving on to the next question. After 24 hours, only 0.125 grams of initial quantity of 1 gram of a radioisotope remains behind. Its half-life period is. So initial quantity is given, final quantity is given, time is given. You have to calculate for the half-life. So radioactivity question. Calculate the half-life for this. Quickly do the calculation and write your answers in the chat box. Yeah, I have one answer in the chat box. What about others? None of you are able to solve it. I have only one answer in the chat box. Quickly do the calculation. We have already discussed how to solve these questions, right? Do you have any doubt in this? Then also let me know. Yeah, I have one more answer in the chat box. And I have two different answers. So anyone else is able to solve this? So after 24 hours, 0.125 grams out of initial quantity of 1 gram of radioisotope is left. Let's see how do we solve this. So first we have to calculate the decay constant. So you know the formula, lambda is equal to 2.303 by T log A by A minus X. So A is the initial quantity, A minus X is the quantity remaining. So quantity remaining is 0.125, initial quantity was 1 gram. Time was 24 hours. So just solve this, you will get the answer as 0.0866 R inverse, and you have to calculate the T half. So if you remember, T half is equal to 0.693 by lambda. So if you have the value of lambda, you can calculate the T half, and if you have the value of T half, you can calculate the value of lambda easily. So when you divide this, you will get the answer as 7.99. So option B is the correct answer. Sneha, you have answered option C. I think you might have done some calculation mistake. Can you check once and let me know? And others who are not able to solve this, is it clear now? Is the calculation clear to everyone? Okay. Okay, if there's no doubt, then I can move to the next question. Which organism is known as an indicator organism in the water treatment? Which microorganism is known as an indicator organism in water treatment? Option A, E. coli, option B, any virus, option C, fungi, and option D, frogs. Which microorganism is known as an indicator organism in the water treatment? Write your answers in the chat box. Okay, I can see 4 of you answered quickly. Option A, E. coli. Anyone else wants to answer? So correct answer is E. coli, Escherichia coli. So it's a I told you in one of the previous session, it is found in our gut. It helps with the digestion of our food. So it is present in our gut. It's a gut microorganism. It is present in our gut. So it is used as an indicator organism in the water treatment because it can actually indicate whether the water is contaminated with feces. Okay, so if there is fecal contamination in the water or not. So since it is present in our gut, it is obviously present in the feces as well. So when the feces are discharged in the water, whether the water after water treatment, whether the fecal contamination has been cleared off from that water or not, that can be used, that can be found out by using this E. coli, by detecting this E. coli. Okay, so E. coli is a very common bacteria that is used in our lab. So detecting it is also easy. There are methods by which you can determine the amount of E. coli present in the water, and based on that amount you can actually tell if the water is potable, if the water is safe for drinking or not, if the how much fecal contamination is present in the water. Okay, is this clear? If anybody has any doubt or any question, please ask. Okay, moving to the next question. dash process is used to destroy all living cells, spores, viruses, and uh, from an object. Dash process is used to destroy all living cells, spores, and viruses from an object. Option A, sterilization. Option B, pasteurization. Option C, antisepsis. And option D, disinfection. What would be the correct answer? Write your answer in the chat box. There are 4 answers in the chat box. All of them are option A, sterilization. That is the correct answer. So, sterilization is the process used to destroy all living cells, spores, and viruses from an object. Other than that, the other 3 options, pasteurization, antisepsis, and disinfection, these are also methods of removing microorganisms. Pasteurization is a process of partial sterilization where you remove the harmful microorganisms by heating the uh, product at a very high temperature. So milk, I have given you the example of milk. It is generally the packaged milk that we purchase from the market. They are generally pasteurized. They are sterilized by doing pasteurization to remove the harmful microorganisms that are present in the milk so that it is safe for us to drink. Then comes antisepsis. Antisepsis is a process of preventing the growth of microorganism on the living tissue. Okay, so whenever we get a cut or wound, we apply some antiseptic solution to prevent any other microorganisms to grow on that wound and to enter our body through the wounded area and cause infection. And disinfection is a process of removing the microorganisms from the surface, inanimate object surfaces. So like we mop the floors at home by using some disinfectant solution in the water, or we wipe the surface of our tables with some disinfectant to remove the pathogens that are present on the surface. Okay, I hope that's clear to everyone. Next question is, vaccine for smallpox was discovered by. The vaccine for smallpox was discovered by option A, Louis Pasteur, option B, Alexander Fleming, option C, Edward Jenner, and option D, John Mendel. Vaccine for smallpox was discovered by. All of you write your answers. Four of you have answered. What about others? Anyone else wants to answer? Who discovered the vaccine for smallpox? Okay, 4 of you are saying option C, Edward Jenner. That is the correct answer. So, Edward Jenner discovered the vaccine for smallpox. So, how did he do this discovery? He actually used the pus of cowpox, which is another similar family virus. So, smallpox is viral disease, and there's another virus that causes cowpox, which is less lethal than smallpox. So, what Edward Jenner did, he took the pus from a patient suffering from cowpox. So all these smallpox, cowpox, even chickenpox, you know that in these diseases we get blisters, and then the blisters are filled with pus. Okay, so rashes and blisters, they are filled with pus. So he took the pus from the cowpox blister of a patient, and then he injected that pus into another patient and saw that that particular patient did not develop smallpox after injecting the cowpox pus. That's how he came up with the idea of vaccination, that if we inject a small dose of either inactivated pathogen, or if we inject a small dose of a pathogen from the same family, our body will recognize that pathogen, and our body will develop an immune system against that pathogen, so that when actually we get the infection from the main pathogen, we won't develop a very serious infection. So, like, you all must be vaccinated with COVID vaccine or polio vaccine. So those vaccination program and those vaccination procedures, they were the beginning of that concept of vaccination was given by Edward Jenner. Okay, so when we were vaccinated by a polio vaccine, when the polio virus entered our body knew that it's a harmful virus and we have to fight against it. And because our body was already immune towards the polio virus, so we did not develop polio. And similarly, COVID also, COVID vaccine was given so that our body now knows that okay, this particular virus is uh, like, our body knows that it has to fight against this particular virus. Okay, so Edward Jenner was the one who developed the concept of vaccination. And these are the major milestones in microbiology. So all the other people that are given in the option, Louis Pasteur, Alexander Fleming, John Mendel, all of them are actually, all of them have contributed in the field of biology in one way or the other. So we have listed their contributions. So Robert Hooke was the one who has first observed a cell under a microscope, and then Leeuwenhoek was the one to observe microbes for the first time. So Leeuwenhoek is also known as the father of microbiology. Then Carolus Linnaeus gave the nomenclature system for organisms. Edward Jenner, I already told you, he gave the first vaccine. Louis Pasteur has multiple discoveries. He was the first to understand the process of fermentation. He disproved the spontaneous generation theory. He also developed the process of pasteurization, which I told you, it's a partial sterilization process. Then Joseph Lister came up with the pro- necessity of aseptic surgery. So during surgery, a lot of patients were dying post surgical infections, by developing post surgical infections, because at that time no one was using any antiseptic liquid on the surgical wounds, and the wounds were developing microbial infection. So Joseph Lister was the one who first developed the process of aseptic surgery, where the use of antiseptic solution was introduced during the surgery to prevent the post surgical infection. Then Robert Koch came up with the germ theory of disease. Vinogradsky demonstrated the sulfur cycle. So we have discussed the Vinogradsky column, how we can perform the experiment ourselves in the lab. Then Fleming, Chain and Florey, they are the one who discovered penicillin. Beadle and Tatum gave the relation between the genes and enzymes, that genes are coding for enzymes. Avery, MacLeod, and McCarthy, they gave the they gave the theory that all the genetic material for all organism is DNA. Watson and Crick came up with the structure of DNA, and Carl Woese came up with the another class of bacteria, that is Archaebacteria. Okay. Moving on to the next question. Anybody has any doubt or any questions? Please let me know. You can write your doubts in the chat box, or you can unmute and ask, whatever is comfortable. Next question is Rhizobium bacteria. Option A helps in digestion. Option B helps in nitrogen fixation. Option C causes disease. And option D all of the above. Rhizobium bacteria. Options are helps in digestion, helps in nitrogen fixation, causes disease, and all of the above. What would be the correct answer? All of you write your answers in the chat box. I have 4 answers already. Anyone else wants to answer? Okay, 4 of you are saying help in nitrogen fixation. That is the correct answer. So I've told you in the previous session only, rhizobium resides in the special structure called nodules in the roots of the leguminous plants. So these leguminous plants, in their roots, there are spherical structures which have these bacteria, rhizobium bacteria, and what it does, they are in a symbiotic relationship with these leguminous bacteria. What do we mean by symbiotic relationship? It means that both the plant, the leguminous plant, and the rhizobium bacteria, both of them are benefiting from each other. How they are doing so? So they are doing so by uh, helping each other. So rhizobium bacteria, they can actually do the nitrogen fixation from the atmosphere. So they can uh, convert the nitrogen in the atmosphere into ammonia, and that ammonia can be used up as a nitrogen source by the plant. So the leguminous plants can absorb the ammonia that is fixed by the rhizobium from the roots, and they can use the ammonia as a nitrogen source. Okay, and in return, what rhizobium is getting? Rhizobium is getting a place to live in, nutrients from the plant. So they are living in the roots, so they can easily get the uh, different carbon sources from the plant, and they are surviving in the plant. So they are giving, so the plant is letting the bacteria live, and bacteria is helping the plant to grow better. Okay, so it's a symbiotic relationship, and the rhizobium bacteria helps in nitrogen fixation. Is this clear? If you have any question or any doubt, please let me know. The next question is, which of the following has an alpha-1,6 glycosidic linkage? Which of the following has an alpha-1,6 glycosidic linkage? Option A, amylose. Option B, cellulose. Option C, glycogen. Option D, glucose. Amylose, cellulose, glycogen, glucose. Which of them has an alpha-1,6 glycosidic linkage? Read the question carefully and then answer. Amylose, cellulose, glycogen, and glucose. I already have 2 answers in the chat box. All of you write your answers in the chat box. Why only 1 or 2 person is writing the answers, I don't know. Okay, I have 4 answers in the chat box. All of them are option C, glycogen. That is the correct answer. So amylose has an alpha-1,4 linkage. So amylose is the linear form of starch. So starch is made of 2 polysaccharide units, amylose and amylopectin. So amylose is the one that has linear chain of glucose with alpha-1,4 linkage. So I've told you what is alpha and beta glucose, and so So depending on the position of the OH in the second carbon, we can, sorry, in the first and the second carbon, depending on the position of OH, if the position of OH in the first and the second carbon is in the same direction, it is an alpha glucose. If it is in the opposite direction, that is, it is in a trans configuration, it is a beta glucose. So an alpha glucose is connected by the first carbon and the fourth carbon, it will form an alpha 1,4 glycosidic linkage. And if a beta glucose is connected by the first carbon and the fourth carbon, it is beta 1,4 glycosidic linkage. Okay, so amylose has a alpha 1,4 glycosidic linkage. Cellulose has a beta 1,4 glycosidic linkage between two glucose molecules. Okay, and glycogen has both alpha 1,4 and alpha 1,6 glycosidic bonds. So glycogen, it's a branched molecule. So the linear part is connected with alpha 1,4 linkage, just like the starch or amylose. But the branching happens from the first and the sixth carbon. So first carbon is connected with the sixth carbon. So here, this is the sixth carbon, right? CH2OH, the top one. So if this is connected with another glucose molecule, that will cause branching of this molecule, and this is how glycogen is formed. So it's a branched molecule. That branching occurs from the alpha 1,6 linkage, and glycogens are the part of cell wall as well as surface receptor molecules. Okay, is this clear? Next question is, a nucleotide is different from a nucleoside due to, a nucleotide is different from a nucleoside due to option A, presence of phosphate group, option B, presence of sugar, option C, presence of nitrogenous base, option D, absence of sugar. Nucleotide is different from a nucleoside due to, what would be the correct answer? All of you write your answers in the chat box quickly. Anyone else wants to answer? A nucleotide is different from nucleoside. I have 4 answers in the chat box. All of them are option A, presence of phosphate group. That is the correct answer. So this is the difference between the nucleotide and nucleoside. So once the phosphate groups are added, it is called a nucleotide. So we have, in a nucleoside, you have the sugar and the nitrogenous base. When the sugar is connected with the phosphate, it becomes a nucleotide. And nitrogen, there are 4 types of nitrogenous bases, mainly 5 types of nitrogenous bases. They are divided into 2 categories, purines and pyrimidines. Purines have adenine and guanine, so it's a double ringed structure, nitrogen containing ring structure, and pyrimidines, it's a single ring structure, six membered ring, and the types of pyrimidine are thymine, cytosine, and uracil. So adenine, once it becomes a nucleoside, it will be adenosine. Okay, and then if it is connected with 3-phosphate group, it would be adenosine triphosphate, that is ATP, which is the energy molecule. Remember, I hope this is clear. If you have any questions, please let me know. Next question is, which phase has the condition of specific growth rate mu is equal to 0? Which phase has the condition of the specific growth rate mu is equal to 0? Option A, lag phase, option B, log phase, option C, stationary phase, option D, death phase. Which phase has the specific growth rate as 0? Lag phase, log phase, stationary phase, death phase. What would be the correct answer? All of you write your answers. I have 3 answers in the chat box. Anyone else wants to answer? Okay, 3 of you are saying option C, stationary phase. So now I have 4 answers. All of you are saying option C, stationary phase. That is the correct answer. So, in a growth curve, I've told you there are 4 phases in a bacterial growth curve. So, we have a lag phase, a log phase, a stationary phase, and then a death phase, right? This is lag, this is log, this is the stationary, and then you have the death phase, right? Now, what do we mean by the specific growth rate? Specific growth rate means what is the rate of growth. So, rate of growth means how many new cells are forming and how many cells are dying. Okay. So, now in a lag phase, you know the cells are preparing to enter the log phase. So, they are increasing in size and they are making new DNA and they are getting adapted to the new uh, media. So, there's not a lot of increase in the cell number, but the specific growth rate will be near to zero but not exactly zero because there is no cell death happening. Okay, so it will be approximately equal to zero, but it would be somewhere more than zero. Okay, it can't be uh, exactly zero because there's some amount of cell division that has begun. Okay, in the log phase the cells are at the maximum the cells are growing at the maximum rate. So mu is maximum at the log phase where cells are constantly dividing and multiplying and they are increasing in number. So they are constantly doubling in number. Then in stationary phase, what happens? The number of cells that are dividing and forming new cells, the new cells, the number of new cells is equivalent to the number of cells that are dying. So, okay, so let's say 2 cells divided, let's say we had 4 cells, okay, from which 2 cells divided to form 2 new cells and 2 cells simultaneously died. So, the change in the number of cell, the change in the number of cell will be constant and hence the specific growth rate if we see that will be 0. Sorry, it got erased, but the specific growth rate at a stationary phase will be 0 because the change in the number of new cells that are coming will be 0. There's no change in the number of cells. Whatever the initial number of cells were there, the final number of cells are also same because how much ever the number new cells are forming, equal number of cells are dying. So the final number of cells remain the same and the specific growth rate is 0 at the stationary phase. In the death phase, it will be because the number of dead cells will increase in number more than the number of live cells. So the cell number, effective cell number will decrease and the death growth rate will also decrease. Okay. Any question or any doubt? Next question is lag phase constitute of option A, no change in number, option B, sorry, no change in number but an increase in mass, option B, change in number but decrease in mass, option C, no change in number and decrease in mass, option D, constant number and mass. Lag phase constitute of All of you write your answers in the chat box. I have 3 answers in the chat box. Anyone else wants to write the answer? Okay. 3 of you are saying option A, no change in number but an increase in mass. That is the correct answer. So lag phase constitute of I told you the lag phase is the phase where the cells are preparing to divide. So they increase in mass, because the genetic material gets doubled and everything, all cell components increase in size so that they can divide into 2 new cells. So the cell number does not really change, but the cell mass increases. So this is the growth curve that I was explaining in the previous question also. And we have already discussed this. So if anyone has any question, please let me know. I hope that's clear. If you have any doubt or any questions, please let me know. Okay. Next question is average size of the cell during exponential phase of growth is option A greater than lag phase, option B lesser than lag phase, option C equal to lag phase, and option D constant. What is the average size of the cell during exponential phase? Write your answers in the chat box. Quickly answer because these are all theoretical questions. Shows the graph in question number 16. That's just the growth curve. This is what you are asking.
No, ma'am, not this question. It is question number 17. I am asking for question number 16.
This one?
Yes, ma'am. Thank you, ma'am.
Yeah. This is the same thing. This is only, so basically this is the stationary phase where your specific growth rate will be 0.
Okay, ma'am.
Answer this question quickly. I have only 2 answers. What will be the average size of the cell during exponential phase? Come on, it's not a difficult question. You don't know the answer or you don't want to answer. Okay. Okay, so all of you are saying option B, lesser than the lag phase. That is the correct answer. So in the previous question itself, I explained that in the lag phase, the cell mass increases. So when it enters the log phase, where cell is actively dividing into 2 cells, the cell mass, average cell mass will be lesser than the lag phase. Okay. Which of the following is an organism that obtains its energy from the transfer of electrons originating from chemical compounds and its carbon from an inorganic source? Obtain its energy from the transfer of electrons originating from a chemical compound and its carbon from an inorganic source. So what would we call that organism? Options are chemoautotroph, chemoheterotroph, photoheterotroph, and photoautotroph. I've explained this earlier also. What would be, how do we classify these organisms? Based on the energy source and based on the carbon source. Four of you have answered option A, chemoautotroph. That is the correct answer. So chemoautotroph, because it is taking the energy from the chemical compound, so chemo, and taking the carbon source from carbon source is the inorganic carbon, so autotroph. Inorganic carbon means CO2. So if the carbon source is CO2, it is autotroph. If the carbon source is organic carbon, it is heterotroph. Light energy, then photoautotroph or photoheterotrophs. Chemical energy, then chemoautotroph or chemoheterotroph. Okay. So this classification is very important to know, so that you can always tell by the by looking at the energy source and the carbon source, you can always tell whether the organism is a photoautotroph, photoheterotroph, chemoautotroph or chemoheterotrophs. Okay. Moving on to the next question now. Which of the following is not a name for the cycle resulting in the conversion of 2 carbon acetyl to 1 ATP, 2 CO2, 1 FADH2 and 3 NADH molecules? Which of the following is not a name for the cycle resulting in the conversion of 2 carbon acetyl to 1 ATP, 2 CO2, 1 FADH2 and 3 NADH molecules? Option A, Krebs cycle, option B, tricarboxylic acid cycle, option C, Calvin cycle, and option D, citric acid cycle. What would be the correct answer? All of you write your answers in the chat box. I can see 4 answers in the chat box. Anyone else wants to answer? Four of you are saying option C, Kelvin cycle. Okay, I have one more answer. So 5 of you are saying option C, Calvin cycle. That is the correct answer. So all 3 names, Krebs cycle, tricarboxylic acid cycle and citric acid cycle, all 3 are the names of the same cycle, which is basically where the acetyl coenzyme is entering, and we are getting all these electron carrying compounds, FADH2 and NADH molecules, that can take the electrons and enter the electron transport chain, which will ultimately result in the ATP generation. Okay, so Calvin cycle is a different cycle. Other than that, all 3 are the names of the same cycle. So this is how TCA or citric acid cycle or Krebs cycle works, where we have the acetyl coenzyme A. So if you remember, in the previous session I told you, the cycle actually starts from, the basic metabolic cycle starts from glycolysis. So you have glycolysis first, then this TCA cycle, and then you have the electron transport chain where you have ATP synthesis, right? So what happens in glycolysis? Glucose gets converted into pyruvate. So 1 molecule of glucose gets converted into 2 molecules of pyruvate. Then this pyruvate gets converted into 2 molecules of acetyl coenzyme A. These 2 acetyl coenzyme A, they will enter the TCA cycle. So acetyl coenzyme A will get attached with oxaloacetate to form a 6 carbon molecule citrate. Acetyl coenzyme A is a 2 carbon molecule, right? So 4 plus 2 will give a 6 carbon molecule citrate. Citrate will get converted into isocitrate, then alpha ketoglutarate. In this reaction, first decarboxylation reaction is happening. Then from alpha ketoglutarate, we'll get another decarboxylation will happen, and we'll get a 4 carbon compound, succinyl coenzyme A. So these 2 are the decarboxylation steps. Then from succinyl coenzyme A, we'll have 1 ATP formation and succinate formation. From succinate, we'll get fumarate. From fumarate, we'll get malate and then oxaloacetate, and again the cycle will continue. The 2 decarboxylation step and the conversion of malate to oxaloacetate, 3 of these steps give us NADH, and succinate to fumarate step give us FADH2. These NADH and FADH2 then enter the electron transport chain. So if you remember the complexes of electron transport chain, we had complex 1 and complex 2. Complex 1 can actually take up the electrons from NADH, and complex 2 can take up the electron from FADH2. Complex 2 is the succinate dehydrogenase itself. Both of them can transfer the electron to complex 3 via cytochrome C, and then you have the complex 4 from where the electrons are accepted by the oxygen molecule or water molecule. And in this electron transport process, a lot of protons are pumped out of the mitochondrial matrix, and that creates a proton gradient. That proton gradient drives the ATP synthesis, ATP synthase enzyme, and we get the ATP as the uh, energy currency. Okay, I hope that's clear. Next question is, the biogeochemical cycle of which of the following elements is based on the changes in solubility rather than redox chemistry? Option A carbon, option B sulfur, option C nitrogen, option D phosphorus. Biogeochemical cycle of which of the following elements is based on changes in solubility rather than the redox chemistry? Write your answers in the chat box, all of you. This is the last question of the session. Please answer this question. Biogeochemical cycle of which of the following element is based on the changes in the solubility rather than redox chemistry? All of you write your answers. I have 3 answers in the chat box. Anyone else wants to answer? Nobody? 3 of you who have answered, they are saying option D, phosphorus. That is the correct answer. So all the other 3 options, carbon, sulfur and nitrogen, they are based on the biogeochemical cycles for these elements. They are based on the redox chemistry, because for nitrogen, you know, the nitrogen gets converted into ammonia, then ammonia gets converted into nitrate, so that's an oxidation process, and then denitrification happens, which is a reduction process. So that's a redox chemistry. Same goes for sulfur. So from hydrogen sulfide we get sulfates, and then from sulfates again we get hydrogen sulfide and elemental sulfur. So again, at multiple oxidation states, this sulfur is present in the environment and it is circulated in the environment. Same goes for carbon. Carbon is present in multiple oxidation state. Carbon can be present as carbon dioxide, having a different oxidation state, and carbon can be present as organic carbon in different oxidation states. Okay, so all of these elements, they are present in multiple oxidation states in the environment, whereas phosphorus, it is mainly and only present in the form of phosphates. So in our body or in the environment itself, it is only present in the form of phosphate, and it gets cycled only based on its solubility. So uh, it's found in the earth's crust, and it is cycled from the earth's crust into the organism and back to the earth's crust by difference in solubility. So wherever it is more soluble, it gets transferred to that part. Okay, but it is always present in phosphate. So you can see in our body also, all those reactions, ATP and everything is present. It is carrying the phosphorus in the form of phosphates. Okay, so this was the last question in our session. uh, Please let me know if you have any questions or any doubt, not just from today's session, but from any other session, since today is the last session for this course. After this, you will directly have your exams. So if you have any doubts in any of the question, please let me know. Or if you want to ask something about any topic. Anyone wants to ask anything? camera now. Any doubt or any questions? So I'll look into the two questions that you mentioned in the start of the session from week 11's assignment. I'll get it rectified. From the week 12 also, I would request if any one of you can mail me uh, if any question, if any answer key is given wrong. I can write my mail ID in the chat box, and then any one of you can contact me. Don't spam my mail, please. This is my mail ID, so one of you can save it and just send me a mail if there's any error in the week 12's answer key as well. I'll look into it and I'll get both the questions rectified. Okay. Anyone else wants to
Okay, ma'am.
ask anything? Okay, then if there's no question or no comments, then I think I can end this session. So thank you everyone for joining all the sessions, and I wish you all the best for your exams. Practice well, practice the numerical questions mainly, because a lot of times people tend to make mistakes in those questions. So prepare well for your exams. If you want to ask me anything, you can still ask. Okay. Okay, then if nobody wants to ask anything, then I can close the session today. Thank you all for joining. All the best for your exams. Do well.
Thank you so much, ma'am.
Yeah. Okay. Bye bye.